Great. Good afternoon, and thank you for joining us at the 45th Annual Canaccord Genuity Growth Conference. I'm John Newman, one of the biotech analysts at the firm. We are extremely excited to have Merus with us today, very high-quality biotech. I predict it is the next mega cap. And with us today, we have the CEO, Dr. Bill Lundberg. Thank you, Bill. Bill, I wonder if I could start off by asking you about how Merus bispecifics, or biparatopics, I should say, are differentiated from some of your competitors.
First of all, thank you, John, and thank you to the entire Canaccord Genuity team for your continued support and the opportunity to be here as well. We appreciate that. We also appreciate that it's right in our backyard, so if you facilitated this for us, that's great. We love that. This is a really exciting time for Merus. Merus is an oncology-focused company with multiple clinical assets of our own and several with our partners and collaborators such as Incyte, Loxo, Gilead, and others. And we've been innovators in the field for more than a decade. There are many different platforms for bispecific and multispecific antibodies, but we think what's really important is that we make our bispecifics and multispecifics to the best of our ability, following the tried and true playbook of how to make monoclonal antibodies. And that's important because we know how to make really good medicines out of monoclonal antibodies. And so following that paradigm, I think what you're seeing from our portfolio is a number of really interesting molecules, in particular petosemtamab. But I'd also remind everyone that last year we had now Bizengri, formerly called zenocutuzumab, approved as well. And we believe this approach of standing on the monoclonal antibody experience of what works is a key to our success and contributes to us getting really good molecules. So, for example, not only the ability to do true high-throughput screens to let the biology select for the best candidate, but to be able to do all the fully human IgG1 engineering for extended half-life, for stealth silencing for T-cell engagers, or for combinations in the world of antibody-drug conjugates. We also have the ability to stand on process development and manufacturing methods and approaches and global CDMO capabilities that are the core bedrock of monoclonal antibody discovery, development, and success. And all of this doesn't guarantee success in the clinic, but as I've said, it allows us to identify and risk mitigate in a systematic way and improve the chances for success for our molecules. And I think that's what we're now finally coming to see with our portfolio.
Okay, great. Thank you, Bill. Regarding petosemtamab, your lead asset, just wondering if you could spend a moment talking about the dual targeting of this molecule and why you think that's important clinically.
So petosemtamab is a bispecific antibody that targets two different cell surface antigens. One is the well-established cancer antigen EGFR, and the other is a novel target in oncology called LGR5. We didn't set out to make a bispecific targeting these two. Instead, because we can rely on the monoclonal antibody type approaches, we asked the question, what can we add in high throughput screens to things targeting the EGFR family to really develop much better activity and cell activity for cancer type cell structures as compared to normal structures, and much better activity above and beyond just EGFR, for example, alone. And what we identified was multiple instances of this particular combination pair, EGFR on one side and LGR5 on the other. LGR5 is a really interesting and important molecule, and I'd refer you to a mechanism of action paper that we have on our website if you want to go into it in more detail. It is involved in cell biology in important ways. It's involved in adult tissue stem cells, particularly in the colon, and it's been very well established as a colon cancer target. Cells that express LGR5 appear to be the bedrock of cell growth and metastases in this tumor. From a mechanism standpoint, LGR5 appears to internalize and associate with the proteasome degrader complex. So in the presence of our antibody grabbing onto both LGR5 and EGFR, we get internalization of that whole complex and degradation and elimination of EGFR from these cells. And then finally, our antibody is ADCC enhanced or optimized for having the patient's own immune system help antagonize or fight the cancer. So we think these are really important elements, particularly not only as monotherapy and in combination with drugs like pembrolizumab, which takes the brakes off the T cells, or systemic chemotherapy, which can combine beneficially with antibodies in this setting.
Okay, thank you, Bill. Could you summarize the clinical data for petosemtamab in head and neck cancer that you presented at ASCO this year? It was a really stunning result, and I just wondered if you could walk through that for us.
At ASCO this year, we reported out updated data on a cohort of patients with head and neck cancer, and this is recurrent metastatic head and neck cancer, who had not previously been treated with any systemic therapy, so first-line therapy. The standard therapy for these patients is typically pembrolizumab or pembrolizumab combination, and we showed with the combination of pembrolizumab and our drug, petosemtamab, really a striking and unprecedented 63% response rate in this population of 43 patients. What was striking about this is that it was not only the response rate with 6 CRs, and 14 of these patients who had achieved response were still continuing on therapy, but that the responses occurred across all major subgroups and populations. So real consistency across the entire data set. The median progression-free survival was 9 months, which again is 3 times longer than what pembrolizumab would have provided alone. And median duration of treatment and overall survival were not yet reached. So we had not reached the medians there. In terms of the safety profile in 45 evaluable patients, the drug was very well tolerated. The only point to make here is that bispecifics that contain EGFR do have infusion-related reactions as a characteristic. And as you can see over time, we've worked assiduously to really reduce that rate of infusion-related reactions and severity of reactions to be manageable at this point, and similar to or better than already approved EGFR bispecifics in this setting. It's important to put this in context also as compared to currently available therapies, where today the approved regimen of pembrolizumab alone only has a response rate of around 19 to 21%, and the median progression-free survival only about 3 months. Pembro plus chemo is maybe a response rate of around 34, 35%, but the progression-free survival isn't substantially improved. And what we have with our regimen, adding our drug petosemtamab to pembro, is a tripling, or against the chemo combo, doubling of this benefit, really a substantial benefit and really unprecedented results. In terms of survival, we didn't reach a median survival, but already at 12 months, around 8 out of 10 patients were still alive. That has never been seen before in a head and neck cancer study of this type. So it's really a dramatic response. And I think the broader community has received that really favorably.
Great, thank you. Next question is, what are the plans for the petosemtamab BLA filing in the second line and a later setting of relapse and metastatic head and neck squamous cell carcinoma? And are you planning on using the accelerated approval pathway here?
So here now we're shifting from the first line data set that I told you about to the second line and beyond, where we have previously shown at a study at a conference at the end of last year, clinical data for our drug alone in head and neck cancer. Here too, the efficacy we've shown with a response rate of 36% in 75 patients and median overall survival of 11.5 months is substantially greater than the therapies that are currently available for patients. And we've guided that we have both a second, third line registration trial ongoing and a first line registration trial. For both of these trials, we believe that a strategy of potential accelerated approval based on an early endpoint, such as overall response rate, is something that's available to us. This is both in direct discussion with the FDA and the FDA's public comments about a Project Frontrunner paradigm, where clinical trials that can consider reaching approval on an early endpoint with confirmation of benefit coming from survival from the same trial to convert that to a regular traditional approval. So we think that is the paradigm under which we're following the registration trial.
Okay, great. And could you describe the design of the potentially confirmatory trial that you have running? And I think you've said you expect near complete enrollment by the end of this year. Just wanted to confirm that.
That's right. To focus on the second, third line registration trial, this is a randomized trial of our drug petosemtamab alone, which is exactly the same model that we have these data in 75 patients, compared to an investigator's choice of currently available therapy. And this follows the model of the most previous two registration trials in this setting in head and neck cancer, randomized against investigator's choice. And we are enrolling the trial quite rapidly. We have guided to be substantially enrolled by the end of this year. And we believe this will fit under the paradigm of Project Frontrunner, with this one trial, the potential for accelerated approval based on an early endpoint and confirmation of benefit from survival from the same trial.
Okay, great. And I guess this might be a challenging question for anybody to answer, but except for the FDA, but how much overall survival data do you think the FDA might want to look at from the confirmatory study as they evaluate the BLA package?
So the FDA has said under the Project Frontrunner paradigm that these types of programs have the potential for approval based on an early endpoint such as ORR, and we've been very clear that ORR is the initial endpoint for us. They've also commented publicly and in their comments to us about the importance of being able to see how survival is going, not a full endpoint, final endpoint readout, and not a stat sig type of benefit in survival, but just understanding if the survival is trending in the right direction or certainly not going the wrong way. There's been no guidance document, there's been no published precedent of already approved studies to provide information about exactly what that threshold is. We have in our plans and proposals with the FDA provided our recommended approach, and we've aligned with the FDA on this approach. But the FDA has not formally guided exactly what they need from a policy or a guidance document standpoint.
Okay, thank you. I wondered if we could talk about the strategy in the frontline setting, which you spoke of earlier. Might it be possible for you to use a similar accelerated approval strategy there?
So that is a really important point, that we're using this accelerated approval strategy in both the second line and the first line trials. And just like the second line trial follows very closely well-established precedent, you'll see this is a recurrent theme, that the more that we can risk mitigate with things that have been done before, we'll prefer to do that. The first line trial follows very closely the most recent successful trial leading to approval in the first line setting, the Keynote 048 trial. Here we're randomizing our drug plus pembrolizumab against pembrolizumab only arm. And based on our conversations directly with the FDA, we believe this too could fall under the potential for accelerated approval based on an early endpoint such as overall response rate, with confirmation coming from benefit from the survival endpoint from the same trial. Importantly, to your previous question, the FDA has also commented in this setting the importance of understanding how is survival going. Is it trending in the right way or certainly not going the wrong way in this context?
Okay, great. And I believe that you have talked a little bit about your interim analysis plans for both of the randomized studies. Just curious as to whether you'll be looking at PFS at those interims, overall survival, both. Just curious there.
Both the first-line registration trial and the second, third-line registration trial have a similar efficacy evaluation structure. The initial endpoint is overall response rate. PFS is not an endpoint that has been allocated alpha or that has been designated as a regulatory endpoint for approval. Obviously, people look at it, we'll look at it, the FDA will want to know what it is. But the initial endpoint is overall response rate. And the ultimate endpoint is survival in these trials.
Okay, great. Petosemtamab seems to work very well in head and neck squamous cell carcinoma overall, and that includes HPV positive and negative patients. I'm wondering, is there any sort of allocation to HPV positive or negative patients in terms of enrollment for the frontline study, or are you just leaving that open?
So you're right. In the data that we've shared so far, petosemtamab has shown responses in 4 out of 8 patients when given in combination with pembrolizumab. And this is higher than the pembrolizumab efficacy overall, around 19-21% in first-line recurrent metastatic head and neck cancer. So we are really encouraged by the efficacy. I will point out that the several past registration trials and those successful approvals all have incorporated all the patients. They haven't subsetted out any HPV positive or negative patients in this context. So head and neck cancer, the overall population, has been the population of interest. We're enrolling these trials, and really consistently in the first-line setting, around 21-22% of patients enrolled have disease that is associated with HPV in the first-line setting, and around 25% in the second-line setting. We would anticipate something quite similar because we have a similar enrollment and recruitment strategy to the first-line KEYNOTE-048 prior trial and the second, third-line, the KEYNOTE-040 trial and CheckMate-141 trials.
Okay, thank you. Question regarding the EGFR H-score. What do the enrollment criteria look like for the pivotal second and third line study in head and neck, the randomized study, versus the single arm phase 2, or is there no difference there?
So the EGFR H-score refers to the extent of expression of the epidermal growth factor receptor on the surface of the cancer cell. Higher score, more expression. And that is the target of one of the arms of our antibody. We've shown data in our presentations that we don't really see a correlation between responses or activity and the level of expression of this biomarker. In our registration trials, both the first-line trial and the second, third-line trial, we're enrolling all comers with respect to this factor because we don't, so far, see any evidence that it impacts whether patients are more or less likely to have a response.
Okay, great. And just in terms of a survival benefit, perhaps in the first-line setting and also the second-line and greater setting, just in your opinion and based on your conversations with physicians, how do they think about a significant improvement there? You've obviously already shown really compelling data at ASCO this year, but how do they think about improvement in survival there?
So first, let me say that recurrent metastatic head and neck cancer is a very difficult disease. If you're a patient who maybe had some local disease and then you develop recurrent metastatic cancer, even before you've gotten any systemic therapy, you have a 50-50 chance of being alive a year later. That's a huge unmet medical need. We have to be able to do better than that. In the KEYNOTE-048 study that led to pembrolizumab approval, that median survival, the midpoint survival, was only 12.3 months. And that actually bested cetuximab chemotherapy, which is around 10.3 months. So that was an improvement, but nowhere near good enough. That gives you a sense already of what physicians say is important. That bump from 10.3 to 12.3 months mattered a lot. Now, we don't know from our data we presented at ASCO where our median is going to be. We hadn't gotten that far in terms of follow-up. But we do know a couple of things. On average, about 50% of the patients treated with pembrolizumab alone in that clinical trial were alive at the 12-month mark. With our data we shared at ASCO, almost 8 out of 10, 79% of patients with our adding petosemtamab to pembro were alive at 12 months. So already we believe we're seeing a substantial signal and really unprecedented in the world of head and neck cancer. And to my mind, that really does speak to the transformational potential of our drug in head and neck cancer.
Okay, great. Thank you. Wondered if we could pivot now to the work that you're doing with petosemtamab in colorectal cancer. I believe you plan to report some data during the second half of this year, 2025. Just curious as to how we should think about petosemtamab in colorectal cancer, especially versus some of the older therapies, for example, Erbitux.
So colorectal cancer isn't as easy as head and neck cancer because it is a more heterogeneous disease. There are patient factors, there are tumor genetics that impact how patients will do. And when you talk about some sort of benchmarks or numbers for the already established drugs, you have to pay very close attention to what is the population you're talking about. In general, in this population, and here it's metastatic colorectal cancer that's genetically wild type with first-line treatment, in general, the responses we've seen from current standard therapies, a cetuximab antibody with chemotherapy, is around 65% in the, for example, FIRE3 study or the PEAK study. But there's a range among multiple different studies, plus or minus 5 or 10% around that. I will say patients with left-sided cancers respond better and do better with cetuximab than right-sided cancers. And so obviously, if your trial that you're looking at has a lot more patients with left-sided cancer, that's going to impact the numbers. So you have to pay very close attention to the details from a benchmarking standpoint. In the second-line setting, if a patient gets cetuximab plus chemotherapy or panitumumab plus chemotherapy, the response rate's around 30 to 35%, plus or minus 5% across a range of studies. And for example, there's an EPIC study that has given us a data point, but there's a range of studies around there as well.
Okay, great. And in terms of the readout, I think you have a number of cohorts looking at, for example, second line plus, I think third line. What types of data should we expect in terms of which cohorts you might present, and then also the efficacy? I think you've previously talked about response rate, but just wanted to confirm that.
That's right. So let me just take a minute to remind everyone, the original dose escalation for this drug was done in colorectal cancer. And there, through all the various different doses, we didn't see any responses. And when we look back at that data set, it was a very late-line therapy, given median fifth-line, heavily pretreated patients, many genetic mutations that would render cetuximab or EGFR antibodies irrelevant. And many of those patients had prior cetuximab and panitumumab, so it was probably naive of us to think that we would see responses. What we're doing now is running cohorts to try to really evaluate this. Genetically wild type, no prior cetuximab in first and second line metastatic colorectal cancer with standard chemotherapy, and then a third line cohort of petosemtamab monotherapy. These are cohorts that are all continuing to enroll now, and we provide a clinical update on these data the second half of this year, and we prefer medical conferences as a forum.
Okay, great. And could you talk a little bit more about the design of the work you're doing in the second line? I believe that's in combination with chemotherapy. I'm wondering if initially that study is going to look to establish the correct dose as a monotherapy and then proceed as combination, or how you're looking at the dose level?
Importantly, for all three cohorts we're running in colorectal cancer, all the patients started with a full dose and full schedule of petosemtamab 1,500 milligrams. This is a dose that we've established through Project Optimus as an FDA-agreed dose in head and neck cancer. Often the FDA allows companies to stick to that dose and schedule, not requiring additional dose work. So we believe all the patients will be treated with what we think may be the appropriate dose, whether combination first and second line or monotherapy in third line.
Okay, great. Thanks. And then last question I have, we've just got a few seconds left, is what are the longer term plans for development outside the United States?
So we believe all of these trials, when they reach the registration phase, need to be designed for the entire international focus, not only the US, but be able to support registration broadly. We think if our drug works well, we really do want to be able to provide our drug and make it available broadly to patients across the globe.
Okay, great. Great. Well, that's all the time we have for today. I wanted to thank everyone in the audience and on the webcast. And also, of course, thank you to Merus. And thank you very much, Bill.
Thank you.